It turns out that regulation is also a critical function of proteins. Proteins are involved in regulating all cellular processes, whether that's DNA synthesis, RNA synthesis, protein synthesis and breakdown, protein function, metabolic reactions, when DNA should be copied, when protein should be digested by a protease, when a protein should be binding to A instead of A prime, when a specific metabolic pathway is turned on and turned off. Bottom line is that proteins regulate almost every cellular reaction and process that occurs. And that regulation occurs in a whole variety of ways. We'll talk about a few now, and we'll talk about more as we move through the course. So one fundamental way in which regulation occurs is if all of these arrows, the black arrows, represent enzymes carrying out particular reactions. If you have enough of an end product, there is a mechanism called feedback inhibition. It is a negative regulatory process, usually by some product or some intermediate in the pathway. So all of the red lines that have lines at the end indicate inhibition in this pathway. So when you get enough methionine, you inhibit the conversion of homoserine to the first step in the methionine pathway. If you have enough isoleucine, you inhibit threonine conversion to the first step going to isoleucine. These are synthetic pathways for amino acids. If you have enough lysine, you inhibit aspartate semialdehyde, and so on and so on. So you can have these complex regulatory pathways. Again, usually some direct product of the pathway. You can also have stimulation. So if you have another pathway, you can have a product of that pathway, and the green arrows indicate stimulation. Let's say that pathway needs something that's down the line in this pathway. Let's say that it needs homoserine. And so the product of pathway 2, because it needs homoserine, will enhance the conversion of aspartate to aspartyl phosphate and hence increase the amount of homoserine that's present. This feedback inhibition stimulation occurs at the level of protein function. We're not altering the levels of any of these proteins. We're only altering their activity, their ability to carry out the reaction, either enhancing it or inhibiting it. So it's important that we're not changing the levels of the enzymes. We're only altering their activity. We will see later that there are ways to actually change levels of enzymes, but feedback inhibition and stimulation is only at the level of activity. The amount of enzyme is not altered. So how do these changes occur? How does something affect the activity of a protein? And that process is called allosteric regulation. Allosteric actually means different allo structure. And it requires that there is some sort of conformational shift or a change in dynamic properties that either decreases if it's negative regulation or increases the activity of the protein. This requires that there be multiple binding sites in a protein. Not only do these proteins that are subject to allosteric regulation bind to their substrates, they often have a second site for binding the regulator. These proteins are usually multi-subunit. They can have same or different subunits, and they are often symmetric in their structure. And the sites that are utilized are able to communicate. That is, you bind the regulator at one site and it impacts the catalytic site, or it impacts binding at a different site. The communication occurs through the structure of the protein, and there are reciprocal relationships between the effects at these sites. So you can have negative inhibition of activity, as we have seen in the pathways, or it can be positive. You can enhance activity. So negative regulators, positive regulators. And a classic example of allostery is actually hemoglobin binding to oxygen. We have two different subunits, and if you remember, we have myoglobin and hemoglobin. Myoglobin is a monomeric protein, although the subunits, as you can see, of myoglobin and hemoglobin shown to the right are very similar. Myoglobin has a hyperbolic curve. That is, you see a smooth curve for the binding of oxygen, and it binds very tightly at low oxygen concentrations. On the other hand, notice the shape of the curve for hemoglobin. It doesn't bind so well at low concentrations, but once you've bound oxygen, it binds better. And once it binds more oxygen, it binds better, and finally it saturates. So it has a positive allosteric response to the binding of oxygen. You bind a little, then you bind more, and with higher affinity. And so this is a classic example of allostery, where you bind to a substrate and it enhances the binding of the next substrate. And this is different than if you have negative regulation, where you bind at a second site and it inhibits the catalysis at the first site. This whole process of communication is known as allostery. But there are other ways to regulate catalytic activity. It turns out that catalytic activity can be regulated by covalently modifying a protein. So you can phosphorylate serine and threonine, introduce a negative charge. That can completely shut down, or it can completely activate a protein. You can acetylate. Lysine, if you acetylate it, it blocks the positive charge on the amino group, changes the interactions that can occur. With methylation, you can actually expand the radius of positive charge for lysine by either mono, di, or trimethylation. So you can regulate what a protein does, whether bind or catalyze, using covalent modification. What this process does is provide a context. So phosphorylation may say, this is happening outside the cell, so inside the cell, these things need to happen. And it is used for very complex signaling pathways in cells. Note that this is different from the control of the actual levels of the enzyme or protein. And so regulation by modification is often used in complex signaling pathways that detect some input, whether it's outside things like survival factors or growth factors or hormones, and translate that inside into a series of generally modifications of proteins to either activate or inactivate them. Or internal signaling that says, okay, we've got enough of this, let's turn that off. It's interesting that many of these complex signaling pathways are involved in specific regulation of DNA expression, in regulating what proteins are produced at what time in response to the overall cellular context. It turns out that we are discovering, largely over the past decade or so, that there are tremendous numbers of interactions between proteins in the cytoplasm. There are many, many proteins and other components. And these proteins interact with one another. Some interact with lots of proteins, some and are called hubs. Some interact with only a few proteins, and some don't interact at all, but there are numerous interactions that form this complex interaction network that you see on the right. And today we are able to identify the interactions and the proteins that form the nodes that actually become specific regulatory points. So if you look at this sort of star shape in the lower, just below the midpoint on the left, you'll see this sort of star. This is a key node in a network. So the state of that protein impacts lots of other proteins. These interactions are absolutely critical to cell function, and they are complicated and confusing, but they are a very exciting area of exploration and understanding. And in fact, they ultimately drive what's happening in the cell. It is these interactions that lead to cell function. Cell biology encompasses understanding these pathways and having insight into which proteins are acting with which other proteins and what that drives in terms of cell function. And there are even books out on protein interaction networks, and this is obviously a very computationally complex area and also an experimentally complex area, and we are growing our capacity to explore these types of complex interactions within the cell. So, how do you imagine cells manage all of these complex reactions and interactions? Is there a central brain that oversees these processes? What actually goes on in the cell?